Ovaries are one of the earliest organs to age. It starts to age in women beginning in their early 30s to mid-30s. And the diagnosis of ovarian aging should be made in conjunction of chronological age as well as certain biomarkers and parameters that reflect the ovarian reserve, which is the number of eggs in the ovaries. So we use tests like anti-Mullerian hormone that's being secreted by the number of eggs from the ovaries. So they're in these structures called follicles and they make anti-Mullerian hormone, a hormone called follicle-stimulating hormone FSH that's made from the pituitary, as well as doing an ultrasound and counting the number of follicles. Again, those are the structures that have egg inside. But alone, these parameters don't tell the full story. The greatest challenge for infertility treatment for women of advanced reproductive age is the egg quality. We're seeing a dramatic decline in the success rates of IVF with increasing age due to decrease in the egg quality or oocyte quality. In my lab, we're using innovative molecular technologies to modulate early folliculogenesis. What that means is that we have these functional units called follicles in the ovaries, and with the cutting-edge assisted reproductive technology, which is IVF, we can only access the latter part of that. What we're doing now is we're trying to access earlier part of it because especially for women of advanced reproductive age, they have tens of thousands of those early follicles in their ovaries that we cannot tap into using the state-of-the-art approaches. So now we're looking at that earliest stages and using innovative molecular technologies such as identifying new small molecules or using gene therapies or using viral vectors to modulate that early part so that we can increase the number of follicles available to stimulate with IVF that will ultimately improve the fertility outcomes. If successful, these approaches can be paradigm shifting of how we think about the infertility treatment. My lab is exploring approaches such as gene therapy, and we are particularly focused on using viral vectors. What we are doing, we are screening various serotypes, so various versions of these viral vectors, and trying to see which one of them can actually transduce or get into the cells of the human ovaries and modulate the gene expression there. And these approaches can help us to again modulate the rate of folliculogenesis that are currently inaccessible to modern treatments, and in the future can even lead to personalized medicine where we can modulate the expression of several genes to improve infertility treatment outcomes. I'm very excited to be in this field because it's at the intersection of the molecular biology, molecular reproductive biology research, and the clinical medicine. It's a very young field. The first IVF baby in the world was born in 1978. First IVF baby in the United States was born in 1981. So it's such a young field, so hungry for innovation, and those of us who are physician scientists at this intersection are very excited to do this cutting-edge research and bring our discoveries to clinic to help our patients.